Hello and welcome, everyone. Thank you for joining us tonight. My name is Cynthia Henson, and I'm from Simon Fraser University, and I'm happy that you're joining us tonight. And just a reminder for everyone coming into the room that we are recording this event. And I just wanted to make sure you all knew. So we'll just give it a minute or so for more people to come into the attendee room. Thank you so much for joining us so promptly. Again, my name is Cynthia Henson, and this is SFU's Café Scientifique, brought to you by the Dean of Science Office over at Simon Fraser University. Thank you for joining us tonight. I see lots of familiar names. Thank you so much for coming in, and a lot of people coming for the first time too. So a warm hello and welcome. Thank you so much for your support. So let's give it maybe half a minute more before we proceed. Again, this is Simon Fraser University's Café Scientifique, and my name is Cynthia Henson, and I will be hosting this event tonight. We welcome all those joining us from beyond the Lower Mainland and beyond BC. Thank you for supporting this discussion series. All right, I have 5:03 on my time, and I guess we better start with this event. Again, thank you so much, and good evening, everyone. As mentioned, my name is Cynthia Henson, and I work at the Dean of Science Office over at Simon Fraser University, and I do outreach and engagement on behalf of the Faculty of Science. This is Café Scientifique. It is a discussion series where we do feature our faculty members from across our different science departments to engage with the public and talk a bit more about their research work and entertain questions from our community partners. So we hold this about 6 or 7 times a year, and we do this by a webinar format to allow people from beyond the Lower Mainland and beyond BC to join us. Because you joined our event tonight, your email addresses have been included in our mailing list, so you should be receiving updates on the next cafes and next faculty events. If you choose to unsubscribe from that list, just reply back to that same email. We're happy to see lots of Café Scientifique regulars, lots of familiar names. Thank you for always supporting this series. And a warm hello and welcome to all those joining us for the first time. It's great to see lots of our community partners and friends from even beyond BC joining us. Let me begin by acknowledging that we are privileged to gather today on the unceded traditional territories of the Squamish, Tsleil-Waututh, Coquitlam, and Musqueam peoples. We thank them for having cared for these lands and waters since time out of mind, and look forward to working with them in partnership as we continue to build this great city together. If you know whose territory you're currently on, please take a moment to reflect on this. And if you don't know whose land you're on, I encourage you to look it up and make yourself familiar with the Indigenous peoples whose land you occupy. Just a few house rules before I turn over the floor to our presenter for tonight. We do have a Q&A box that you have access to, and we will try our best to read out everyone's questions and comments after the presentation. The chat page is turned off just to keep communication simpler, so just focus on the Q&A box for now. The live transcript has been turned on. You may access that as well for your convenience if that is helpful for you. And just a reminder to all that this event is being recorded, and we will be sharing the recording of this event in our faculty YouTube channel after tonight's event. All right, so as you see in your opening slide, we do have one of our faculty members from our Department of Molecular Biology and Biochemistry. I'm very pleased that Dr. Dustin King has agreed to present for us tonight. And his presentation is actually entitled Decoding How Life Senses and Responds to Carbon Dioxide Gas. It's a discussion tonight of his indigenous background, and he will be delving into indigenous ways of knowing and learning and embracing a two-eyed seeing approach and how this is applied into his carbon dioxide biochemistry work. So I'm very interested in learning more about this, and I hope you are too. So at this point, I'd like to turn over the floor to Dustin and invite him to screen share his presentation for tonight. Good evening, Dustin.
Good evening, Cynthia, and thank you so much for the invitation. I'm just going to take one quick moment here to pull up my slides.
Sure.
Just give me 1 second. All right, so hopefully everybody can see that.
Yes, it is working well.
Just going to move that. Okay, great. Yeah, so thank you, Cynthia, once again for the kind introduction and the invitation to come here and do this Café Scientifique. I think this is just an excellent opportunity for community outreach and for having the opportunity to explain a little bit about what we do in the research community. And so please, feel free to ask me questions throughout the talk, and I look forward to engaging with everybody. So that being said, my research area is really all about how organisms sense and respond to carbon dioxide gas. And it may surprise you that this is an area that we still know relatively little about. So I hope to get into that today and to convince you that this is an important area of research. But before we do, I think it's important to acknowledge, of course, that we're on the traditional territories of the Coast Salish, including the Musqueam, Squamish, Tsleil-Waututh, and Coquitlam nations. And I feel very privileged to live on these lands. And the Coast Salish have been amazing custodians of these lands for literally thousands of years. And I think in the context where we're facing a global climate crisis, it's so important to reflect on how we interact with the land. And I think that our Indigenous communities are great stewards and can really lead us toward developing a more sustainable approach to our interaction with it. So with that, that's going to be part of what the talk here today is about. So I am actually half Ojibwe, Anishinaabe on my father's side. I belong to the Thessalon First Nation, which is a very small First Nations band on the northern shores of Lake Huron in Ontario. The closest city is Sault Ste. Marie, and the Thessalon is literally located right where Lake Superior and Lake Huron meet. So it's my great privilege to come from this cultural background, which my dad really helped to foster for my sisters and I growing up. But I did not grow up living on the Thessalon. I actually grew up in northern BC in a very small town called Burns Lake. And here really took part in the local Indigenous community in these areas, which is the Carrier First Nations, the Tilkako and Wet'suwet'en First Nations, and was involved in those communities growing up as a kid. And really through this lens and through interacting with my father, it really helped me to gain a deep appreciation of nature. And the way that that this really drove me in my professional career is through having a deep fascination with just simply what life is and how it works and how nature works. And this actually drove me to thinking about things in the molecular sense. How does nature at the subatomic or at the atomic and molecular level actually function? And so this drove me to do a bachelor's degree in biochemistry at the University of Northern BC. And I later went on to do a PhD in antimicrobial resistance in the lab of Natalie Stranadka at UBC. And finally on to my independent faculty position here at Simon Fraser University. And all throughout this training, I've had amazing colleagues and mentors, but it's quite striking that I've had no colleagues or mentors that have similarly been Indigenous up until this time. And I find this in a way quite strange, because the very thing that brought me into scientific research, in a way, within the academic community is lacking. And I think that there's so much that can be gained from really combining Western knowledge and Indigenous knowledges together. And I hope that we can move toward that in the scientific community more and more as we proceed. And so it turns out that my experience in academia is not unique in any sort of way. In fact, in Canada, there are about 1.7 million Indigenous people living on our lands. And of those, only approximately 7% are attending Western universities, and less than 1% are within the sciences, even fewer within the biochemical sciences, and a very small proportion are actually in leadership roles. And this is really a shame, in part because, recently within the scientific community, through amazing work done by Victoria Tolley-Carpez and colleagues, we now understand that Indigenous communities are protecting about 40% of the forests on our planet. And this corresponds to about 80% of its biodiversity. In total, these forests are sequestering about 8 billion tons of carbon dioxide every single year. And that's about equivalent to the emissions by the United States on an annual basis. So a significant amount of carbon is being captured and protected by these forests that are basically stewarded by Indigenous communities. Within my own community, the Thessalon, climate change has had a major impact. And I think that this sustainable way of thinking is interwoven into Indigenous culture, in part because Indigenous culture relies so heavily on an interaction with the environment. Within my community, global warming has really resulted in a lot of cyanobacterial blooms within the fresh waterways that are responsible for our major food source, which is manomin, this wild rice. And so through that, we've really seen directly the impacts that climate change has had within our local communities. And with that really comes, one of the main teachings in Ojibwe culture and in many First Nations cultures is the idea that we should seek to protect the earth, carrying forward 7 generations into the future. And so that's an amazing level of sustainability and really a shift in perspective, to not only try to sustain the planet for our children, but to try to sustain it for 7 generations into the future. Now, this may be in many ways quite intangible, but in part, this is my driving force behind what I choose to focus on in biochemistry. And in my work, I'm seeking to understand some of the molecular mechanisms that underlie CO2, and this has implications in climate change. And so why is this? Why am I interested in CO2 biology? This is in part because the general scientific community has now reached a widespread consensus that we've entered the epoch of the Anthropocene. And that's simply the idea that anthropogenic means are a main driver of climate change on our planet. This story really began at the onset of the Industrial Revolution and is part driven by an overuse and a misuse of our technologies in order to generate these industrial processes that, while benefiting temporarily our communities to grow and sustain, ultimately they're pumping loads of carbon dioxide gas into the atmosphere, which has a warming effect. In essence, this CO2, at the time that I started this CO2 research, was at levels that we hadn't seen in almost 1 million years, at levels of about 405 parts per million in our atmosphere. And throughout the previous ice ages, you can see these cycles going to about 1 million years ago, where it never reached these sort of levels. But since giving these talks just a few short years ago, the levels of carbon dioxide are on a runaway, and we can see that they've now increased to about 420 parts per million, really showing in quite a real way how over even a few years, the levels of CO2 in our atmosphere are rising. And this CO2 is a greenhouse gas and has led in large part to a change in the global temperatures since the onset of the Industrial Revolution. And of course, the Paris Climate Accord set this benchmark of trying to maintain global warming and temperatures below 1.5 degrees Celsius. However, it's very clear that within the next 10 years, we're going to be crossing that barrier and entering into very dangerous zones where we're above the 1.5 degrees Celsius benchmark set out in the Paris Accord. And so with that, like, why is CO2 contributing to the warming of our planet? In part, this is because our planet is warmed largely by radiation coming from the sun. And in essence, those photons impinge upon our planet and heat up the core of heat up the surface of our planet. And a large amount of those photons is actually reflected back out into space. But of course, surrounding our atmosphere and the air that we breathe are a number of different gases, among them carbon dioxide, which is a greenhouse gas that can absorb some of that reflected gas and re-emit it really in all directions. But one of these directions that it emits it is right back toward Earth. And this results in a warming of the planet. And this is actually very important for our atmosphere. In fact, if we didn't have carbon dioxide, the planet would be about 30 degrees Celsius cooler and would be largely incompatible with much of the life that we have on the planet. So the greenhouse gas effect is a very valuable thing. Like, this is what we need for our planet. The problem is when we start to tip the scales and produce too much CO2, so this greenhouse effect becomes larger and larger. And if we look at various greenhouse gases, CO2 really is the main contributor within our atmosphere, leading to increased greenhouse gas effect. In fact, it's about 80% of the greenhouse gas effect is coming from carbon dioxide. So what are the things that regulate CO2 within our atmosphere? This is called the global carbon cycle. And in essence, CO2 within our atmosphere is taken up by plants and converted into simple sugars that serve as a sort of chemical fuel for the entire biosphere. This process is coupled to the absorption of sunlight that provides energy in a process called photosynthesis. These plants are then eaten by various forms of life and all types of animals that take up the sugars that were created by the plants originating from CO2, and they themselves eat those plants, and then these respiring organisms breathe out CO2, much like we do. And this CO2 re-enters the atmosphere. Now, these are natural processes. Of course, plants and animals eventually, when we die and when plants die, decompose and end up forming fossil fuels, or a large long-term form of carbon storage. The problem comes when we start extracting these fossil fuels and using them in industry for various processes as an energy source. Ultimately, these sources end up pumping CO2 back out into the atmosphere. And it's this balance that's really driving anthropogenic or human-caused climate change. And so the question is, like, how exactly are we able to reduce our emissions? And this is a multifactorial problem that comes down to the way that each one of us lives, the types of energy that we're using. But I would contend that part of the answer actually comes from changing our processes that we use in industry. And perhaps we have something to learn from how nature actually captures carbon dioxide and transforms it. And this is at the heart of my research, is trying to discover the basic biochemical mechanisms in which life actually does this. And so in order to really begin this story and understand how organisms interact with CO2, it's important to note that CO2 has been present in our atmosphere over the full duration of evolution on Earth. Basically, just after formation of the Earth, nearly 4.5 billion years ago, CO2 levels were incredibly high, much higher than they are today, in fact. Of course, this state of the planet was inhospitable to life forms. And it took, over 1 billion years to reach the right conditions where we started to form the first photosynthetic bacteria that capture sunlight and use CO2 as their main carbon source. These early photosynthetic bacteria produced oxygen as a byproduct and started basically exhaling oxygen all over the planet. These photosynthetic bacteria were in fact so successful and produced so much oxygen that they changed the composition of the Earth's atmosphere and basically created this oxygen-rich environment. Now, at the time, oxygen was toxic to most of the organisms on the planet. And this was behind the greatest mass extinction in our planet's history, the Great Oxidation Event. But through this bottleneck and through this evolutionary pressure came the emergence of organisms that could flip the system on its head. And instead of using carbon dioxide as a chemical fuel, in fact, use oxygen and undergo aerobic respiration, much like we do. And so this led to the onset of the very first green algae eukaryotes that can do this, leading to modern day times where we have many eukaryotic organisms that are able to use oxygen and undergo oxygen-based metabolism. And we see today that we have much higher levels of oxygen within our atmosphere than carbon dioxide. Although, as I mentioned, carbon dioxide is starting to rise again. So in this context, this really sets the stage for the fact that yes, our atmospheric CO2 levels are rising, but low compared to at the start, where they were at the start of the planet. But basically, the environments in which organisms typically thrive are enriched in carbon dioxide. Look no further than us, for example. Within our tissues, due to this aerobic respiration, carbon dioxide is produced in mass. And the average adult human actually exhales about 1 kilogram of the stuff every single day. Within the deep sea hydrothermal vents, these are areas that are teeming with microbial life. And these hydrothermal vents are known to pump out about 90% CO2. So incredibly high CO2 amounts. Within proximity to decaying organic matter, CO2 levels are very high because they're being released as that organic material decays. And what's further is that within each of these contexts, CO2 levels are constantly fluctuating. They vary in their local concentration greatly. And so what this has resulted in is that organisms from every domain on the tree of life have evolved adaptive physiological, biological responses to detect CO2 levels and elicit important biological change. What I mean by that is that they are detecting different levels of CO2 and eliciting a different response, whether you have high CO2 or whether you have low CO2. So let's take a look at a few of those examples and how organisms are using this carbon dioxide. CO2 is really important actually in predator-prey relationships. Look, for example, at the far left at this mouse. Now the mouse, a major portion of its brain is used in smell. It's called the olfactory lobe in its brain. And a major region within this olfactory lobe is dedicated to carbon dioxide sensing. So it can detect CO2 levels that are slightly above atmospheric and recognize that there must be a respiring predator, such as this cat, that is really close to the mouse. So this is used in order to avoid predation by the mouse. Same thing in fish, actually. So both fish seek out their prey based on carbon dioxide gradients, and also the prey runs away from predators based upon CO2 that is emitted. And Drosophila, the fruit fly that we commonly use in the lab, also has a very acute sensing of CO2 in order to avoid predation. On the flip side, the mosquito that we're all familiar with actually seeks us out in part based upon the carbon dioxide that we emit. And this is done, as for many different insects, using specialized neurons. And in the case of the mosquito, you see this proboscis, that's the main thing that is used to suck the blood out of the human prey. Right beside that are these things called maxillary palps. And within these maxillary pulps are these sensory neurons that have a CO2 receptor and are very sensitive at detecting CO2. And this is what essentially is detected by the mosquito that tells the mosquito brain, oh, we have potential prey nearby, let's go there and do our thing. So that's part of the ways in which CO2 is actually used in predator-prey relationships. But of course, within us, CO2 is very important for human health and disease. If we actually look in our tissues, as I mentioned, CO2 is being produced in mass. And that CO2 is basically transported into our blood vessels and transported from our tissues into the lungs, where we exhale it in exchange for oxygen. And then oxygen is delivered to our tissues. Oxygen is used, of course, in this oxidative metabolism, and carbon dioxide as a waste product needs to be removed from the body. Now, CO2 levels are really important within our bodies because they actually regulate blood pH and basically blood acidity. And dysregulation of these CO2 levels leads to basically disorders that we call hypercapnia and hypocapnia. And now CO2 can also have impact on inflammation and innate immunity. So it's very important in human health. So as humans, how do we regulate CO2 levels in our blood? We do this through dedicated receptors that exist within our central brainstem that actually detect CO2 within the blood and undergo an automatic response that causes us to change the rate and depth at which we breathe. This is why when you start to exercise, for example, and your tissues are working really hard, producing a ton of CO2, your brain detects that and tells your lungs to start breathing heavily. And that happens without us even paying attention. So this is very, very important for human health and disease. Not the least of which also is to zoom out and look at how plants, and in particular in relation to the global carbon cycle, use and interact with carbon dioxide. Now, basically, plants undergo photosynthesis, and basically they're able to capture carbon dioxide from the atmosphere and use it to build simple sugar molecules that serve as a sort of chemical fuel that ultimately sustains the biosphere. Now, what we often tend to forget is that photosynthesizing organisms are not only our plants. In fact, over half of the biological carbon fixation on planet Earth occurs in our oceans. And this is due to the phytoplankton that exists within our oceans. Often these single-cell organisms that, similar to plants, use very similar mechanisms to capture carbon dioxide from the atmosphere and build sugars. Let's take a look at one of these organisms. This is actually a cutout of a particular type of phytoplankton that's called cyanobacteria. These are the ones that I mentioned caused the great oxidation event in our early atmosphere. Now, these cells are basically specialized to capture light. You see these stacks of concentric pancakes? This is basically where the complexes are that harness energy from light. And within these circular blue organelles, that's where carbon dioxide is being captured. And if we look over here on the right, these cells are absolutely amazing because they can regulate the pool of carbon dioxide inside the cells. They actually use these particular pumps that are located near the outside of the cell that pump carbon dioxide and its hydrated form, bicarbonate, into the cell and then transport it into these specialized organelles where the sugars are made. So really they have this intricate system to pull CO2 from the atmosphere and use it as a building block. Of course, biological carbon capture has great potential in industry. Currently, the carbon capture mechanisms that are mainly being used in industry are mechanisms that use very harsh solvents and, in essence, capture carbon dioxide from industrial processes and create what we call supercritical CO2, or a liquid form of CO2 that then people take and pump down into basically oil drill sites that are in geologic forms deep beneath the Earth's surface. And the idea is that maybe if we can pump carbon dioxide that was captured in industrial processes deep into these geologic forms, we can prevent it from entering the atmosphere and basically store it underground. And this is what is currently being done. Part of the problem, of course, is, as you can imagine, this CO2 eventually leaks out and re-enters the atmosphere. And what's more is, of course, this requires very harsh solvents that are, in essence, bad for the environment and bad for our health, many of them carcinogens. But what's more is that a lot of oil and gas companies realized that when you do this, you can actually take an oil drill site and use this supercritical CO2 in order to extract more crude oil out of these drill sites. So in effect, they're being used in a way that is actually promoting increased consumption of fossil fuels. And so this is a major problem that we need to try to overcome, especially in incentives for some of these oil companies and switching the way that we do things a bit. And that's part of the motivation for biological carbon capture systems that are really starting to take off. And we can use for this our friend, the cyanobacteria, where we can actually take CO2 produced through industrial processes and divert it over into cyanobacterial holding tanks that can capture that CO2 and convert it into useful forms of molecules that we can use in humanity. This includes, for example, different types of biofuels. It includes using cyanobacteria to create dyes, to create various food products such as spirulina that's really taking off in the food industry, and many different things. So this is one way, a very sustainable way of thinking that can not only capture carbon, but convert it into something that's very useful in the process. So despite all of these varied roles that CO2 plays in nature, and despite its potential implications for human health and well-being and the well-being of our planet, it might surprise you that the very basic fundamental mechanisms through which cells and organisms are actually detecting CO2 at the molecular scale are very poorly understood. And this is at the heart of my research. I'm a biochemist who is really tackling this problem from a biochemical perspective and trying to understand how organisms are doing this. And so when we think about how organisms might be doing this, the most logical place to look is for how CO2 might regulate proteins. And why are we so interested in proteins? This is because proteins are the molecules that perform the vast majority of functions within your cells or within life as a whole. And so there you can really think of proteins as being the workhorses within your cell that are responsible for almost all functions performed by it. So what we're really asking is, how does CO2 regulate proteins? Now, when we think about how CO2 might regulate proteins, the reason why it has been so difficult to understand these pathways is simply because when we put carbon dioxide into aqueous solutions, which all of life is present in aqueous solutions, CO2 doesn't just stay put. In fact, it rapidly reacts with water, and it forms bicarbonate over here on the right, and also gives off a proton. Now, bicarbonate is basically baking soda. So baking soda and this proton are in equilibrium with CO2 in the water. And it can be any of these four species being sensed or detected by organisms. It's unlikely to be water or the proton for various reasons. But when you think about this equilibrium, this is actually why when we have increased atmospheric CO2, it results in acidifying our oceans, because dissolved CO2 gives off this proton. And of course, this is behind coral bleaching and various effects in our aquatic ecosystems. But thinking about how life interacts, there's really two main species through which CO2 might actually be sensed. And that is through the bicarbonate, through the baking soda. And there's been a lot of excellent work done in the scientific community to show certain circumstances where that is indeed the case. But the central view of my lab and my contention is that CO2 itself can play a direct role. And this is kind of, against a lot of thinking within the field. But what is currently underplayed is the fact that CO2 itself is a very reactive molecule that can combine with these proteins. And what I mean by that is that us and others have now shown that at high CO2 levels, particular protein sites are actually able to combine with CO2. And when CO2 is bound, this represents a major change to the character of these proteins. It changes their function. In essence, you can think of it like it can serve as a sort of switch in order to change a protein's role within the cell to something new. And so what's more is that this modification doesn't just stay put, it's actually rapidly reversible. And what I mean by that is if we go from high CO2 levels into low CO2, this modification can revert. And this is important because this is a fundamental principle that allows it to serve as a mechanism to detect and sense CO2. That's because when you have high CO2 levels, you get more of this modification. And when you have low CO2 levels, it reverts and you lose it. So these really, in my opinion, are the hallmarks of a mechanism that might be used in CO2 sensing. Now, in the scientific literature, we only understand a few instances where this is well characterized, but these instances are super important for life. Over here on the left, we can see circled a region where CO2 is bound to the protein responsible for carrying oxygen in your blood, the protein called hemoglobin. And in fact, it regulates hemoglobin binding to this protein, and hence is very, very important for our gas transport in our cells. In plants, such as Rubisco, CO2 binds to this particular protein that is involved in actually fixing carbon dioxide, and it plays a very important role in that process. And we now understand that different antibiotic resistance factors, such as oxa-beta-lactamases, use CO2 modified proteins in order to basically destroy our antibiotics. And this leads to a major problem in clinical medicine. And so despite these few examples, we haven't recognized this modification on other examples in nature. And so why is that exactly? Why do we have a limited understanding of this modification and how it is involved in CO2 sensing? It's basically because the interaction with CO2 is reversible. So as soon as we try to basically purify out CO2 bound to a protein, it's rapidly lost. And we lose that information and are unable to detect the site because it comes on and off. And there are no methods that we can use in the lab to actually detect it. So this is exactly what I set out to do, is to try to develop a method that could be used to actually detect it, and played a bit of a trick in doing so. What we do is to actually use a permanent probe that binds to the site that CO2 would, and it binds so tightly that it is virtually irreversible. It never comes off of these sites. And in essence, what we can do is apply a competition strategy, compete these molecules, where we either incubate with this probe by itself to get the permanent modification, or incubate with the probe plus carbon dioxide. And in the instance where we incubate it with carbon dioxide present, CO2 is able to compete for binding to the site with the probe, and hence block the probe from reacting. And we're now able to detect the difference in these species that have reacted with the probe between the plus and minus CO2. So we're detecting it through competition indirectly. And this strategy is really the nuts and bolts of what my group is currently working on, is developing new strategies to improve these methodologies and reveal more and more CO2 modification sites and go in to study the functional ramifications that this has on cells. But it's really, in our initial studies, we've chosen to focus on cyanobacteria due to their potential in sustainable biotechnology, because of the reasons outlined. And so these are super efficient organisms to work with in the lab. And what has been quite striking so far in our studies is that we've recognized that this modification is not some limited modification. In fact, it is abundant in nature, and it's widespread on the proteins within our cells. And so we're very excited at our future area of research, which is getting more and more into what the functional roles of this CO2 actually are. And so far, we've characterized a few very interesting sites. This is a protein in cyanobacteria that is called P2. And we've recognized that CO2 is able to actually bind in 3 places to the P2 protein. And this has major ramifications because this P2 protein regulates the physiology and the basic metabolism of cyanobacteria. And so we're currently studying what the final effects on the cell might be of this pathway, but it's quite intriguing that CO2 is modulating this regulator of central metabolism. So with that being said, I hope that in the talk today, I've convinced you that studying how organisms sense and respond to carbon dioxide is a very important area of research, and have given you a little bit of insight into some of the core challenges faced by the field, and one of the molecular mechanisms through binding to proteins that this sensing might occur. And really the thesis of my lab and what we're working on is trying to understand how life detects CO2 so that potentially we can learn from it in order to develop more sustainable biotechnologies and more sustainable practices. With that, I'd like to thank my new research group, the King Lab, where we have 3 graduate students pictured over here on the right with me. It's been a lot of fun to start up the lab over the last couple of years with these great folks. To all the undergraduate students that are also doing some research work in my lab, I'd like to thank the key cast of collaborators shown over here on the left, including David Bacadlo and Amy Lee, who also work here at SFU, as well as Alison Smith and Rob Campbell, working at Cambridge and the University of Tokyo. I'd also like to thank my Indigenous band, the Thessalon First Nation, as well as the Wet'suwet'en and Tilkako, the Carrier First Nations on whose lands I grew up. I would also like to thank all of the funding sources that support my new research program, and all of you for your time and attention. I'd be happy to address any questions.
Thank you so much, Dustin. And it's also great to at least meet the rest of your lab team. Very interesting to thank you for including that photo.
Yeah,
so
thanks.
I see a few questions now on the Q&A box, and I thank you so much for typing and sending those in. I do invite everyone else to keep those questions coming. I will try to read through some of them now. And let me start with this one. Can microbiology become a weapon? For instance, in a book that I know, microbiology and bacteria had been developed and could be used together. The bacteria and amoebas could be used to attach others or also be used as identifications. Can you comment on this?
Yeah, that's a great question, actually. So yes, microorganisms can be used as weapons. And in fact, certain microorganisms, such as Bacillus anthracis, the bacterium that causes anthrax, are at the top of the list for potential bioterrorism threats. Some organisms, such as Bacillus anthracis, produce really harmful toxins that if even a few of these toxins get into our system and these bacteria start to grow, they can become quite harmful. So the long story short, even the cyanobacteria that I showed, there are certain species of cyanobacteria that proliferate in our waterways and become very toxic because they produce some of these toxins that I mentioned. And that's why, of course, for certain communities, such as the Thessalon, the wild rice is basically not being consumed because of fear of these bacterial toxins produced by the cyanobacteria. So yeah, great question.
Interesting. Thank you for that. Next question. What caused the major inorganic decrease in CO2 prior to the photosynthetic bacteria evolution?
Oh, I see. So what caused the decrease in atmospheric CO2 levels over time?
Right.
Yeah, great question. So I think that it's in large part due to differences in atmospheric composition and how this has changed over time. But also, you got to consider that in this early environment, photosynthetic bacteria enjoyed having this unique niche where they could simply proliferate all over Earth's oceans. So as they started to do this and became so abundant that they produced a lot of oxygen, they started to change the composition of the atmosphere. And in doing that, they promoted other oxygen-using organisms to come in and fill that niche. So their levels started to decline. So cyanobacteria have a key role in regulating the atmospheric CO2. And when they start to decline, so did the levels of CO2 in the atmosphere.
Right. So it's all related. Well, there's a follow-up question from the same person who asked that question. Is there a similar process of protein binding with H2S in some anaerobic organisms?
Yeah, no, that's a great question because H2S is a very important gas that's used by certain anaerobic organisms. And the answer is basically yes. A lot of those pathways are also not completely understood at this point. And this whole area of gas sensing is quite a challenge because oftentimes these modifications with proteins are reversible and really hard to detect. So long story short, yes, totally.
I like this next question. I'm learning about this in school. Do you have a good way to remember the different types of bacteria?
Oh, yeah. So I think that the thing is that a lot of things when they're being first taught in school, they are taught in a way of memorizing, trying to memorize the bacteria. What I would say is try to get a flavor of what those bacteria are actually doing. Like in the case, if you're trying to remember the cyanobacteria, think about what they're doing in the carbon cycle, and that'll be an easier way for you to grasp onto and actually understand that organism. Same with all types of bacteria. They have interesting characters and things that they do. I would just look into what their major implications are, and that'll be a better way to learn.
Yeah. Right. Just to make more sense out of it, right? And help us remember things.
Exactly.
Thank you.
Yeah.
Next question. Where did the extreme high levels of CO2 in the early Earth come from, and what brought the levels down before the rise of photosynthesis?
Oh, yeah, that's a great question. I think that, in all honesty, I'm not a climate expert, but in the early atmosphere, my understanding is that there was a lot more, the mantle was a lot more dynamic. And so you had a lot more volcanic events that would pump huge amounts of CO2 into the early atmosphere. And that's a major reason for the really high early levels, is because these events were much more common than they are nowadays.
Thank you. Next question. This is my first exposure to indigenous ways of knowing and a two-eyed seeing approach. While this is not your main topic, I would appreciate maybe a short description of what this really means.
Oh, yeah, that's a great question. So the two-eyed seeing approach is a really amazing strategy that was really brought forward into popular knowledge by Dr. Barry Marshall, who's a Mi'kmaq elder, and just an amazing character and a Mi'kmaq elder. And it really reflects a teaching in his culture that was basically used for conflict resolution, is my understanding of it. And basically, the idea is, if we're going to come to some sort of a common ground, we need to understand each other's perspectives. And so the two-eye seeing approach is basically, in literal sense, is trying to see from one eye with one perspective, with your own perspective, and with another eye from another perspective. And I think that this is a very effective strategy for looking at Western science and indigenous science, because if you can gain an idea about what ways of thinking and knowing are important from different frames of mind, we can really enrich our own understanding on a topic. And so that's how I see the two-eye seeing approach.
interesting. And thank you for clarifying that. It's really enriching how to learn, you know, something from different perspectives and trying to understand it in a better way. So thank you for that. Next question. I learned in first aid that the body normally regulates the breathing rates based on serum concentrations of CO2, but that is suppressed in COPD patients in favor of oxygen saturation. What sort of atmospheric CO2 levels would produce this COPD effect in healthy people?
Okay. Yeah, I see. Interesting question about a medical condition, chronic
Right.
obstructive pulmonary disease, which ultimately affects your ability to exchange gas. I think the first thing to note is, of course, that the sensor in our central brainstem that regulates our breathing based on CO2 is not based on atmospheric CO2 levels. It's based on our internal CO2 levels. So because the sensor is in our brain, it's actually sensing the CO2 that is in our blood, which is the CO2 being produced in our tissues. Now, normally, once we get very high CO2 levels in our blood, our brain triggers us to start breathing quite heavily and exhaling that CO2. But in COPD, basically you reach this state where your lungs are not able to efficiently exchange the air, and you have something that's called submissive hypercapnia. So submissive hypercapnia means basically allowing CO2 levels to build up in your blood. And the reason why that happens, my understanding in COPD, is because it would be a waste of our limited resources when we're unhealthy to try to breathe really fast when we're not exchanging air efficiently. And that's why CO2 levels start to build up. But a very complex and good question, but just remember, it's not the atmospheric levels, it's the levels that are in our body that triggers our brain to do that.
Right, right. Next question. Why are marginal increases in atmospheric CO2, even if anthropogenic, is being mistaken for an existential threat?
Oh, I see. I think that yeah, so what the question is probably referring to is the idea that there are many different natural processes that put CO2 out into our atmosphere. Why is it the anthropogenic ones that are the driver of climate change?
Right.
And the idea is that there's a natural balance in CO2. You know, you have volcanic eruptions, or you have various processes naturally that occur in order to release CO2, respiration, for example. But this is being balanced in the ledger of CO2 with these photosynthetic processes that are capturing carbon dioxide at an equal rate. What is essentially happening through our activity is we're putting out extra CO2 above and beyond the natural processes so that we're tipping the levels higher than they are under normal conditions. And this can result in these slight increases in the global temperature, which have major ramifications for life on our planet. So in essence, yes, despite the fact that there are other natural processes that basically give off CO2, think about it more in terms of it's a ledger and human activity is bumping it out of whack.
Thank you. I'll continue to read the questions just for the audience to know. Some questions I'm reading were sent to us in advance before tonight's event. So for future events, if you have questions that you'd like to send in before an event actually happens, because we do send out information about the topics, please feel free to send that by email. So I'll continue to read some of the other questions sent in advance. So someone's asking, I'm curious about the possible reaction of different insects to CO2. A beekeeper once told me that she tries to hold her breath, or at least tries not to exhale much when working with her bees because they seem irritated by CO2. Wondering if this is a more universal insect response, and why is this the case?
Yeah, great question. So, you know, you brought up the example of bees. That's something that we didn't talk about in the discussion, but they also sense these gradients of CO2. And if you look across the fly, the bee, the mosquito, they have very similar sensory neuron systems that are set up to detect CO2 that are basically related to each other through evolutionary history, but they've been repurposed for different functions. In the bee, as you mentioned, this might be a signal that it uses to interpret in its brain, oh, there's some animal close by, I need to defend the nest. In mosquito, it actually uses it to seek out its prey. And in the fruit fly, it uses it to run away from predators. So it's repurposing the same kind of sensory mechanisms for different purposes. But great question.
Right. And I love those examples that you had given on the predator-prey relationships. Very, very interesting. Next question. Given that your research is focused on actualizing a sustainable future, have you adopted any practices to reduce the carbon footprint of your lab?
Oh, yeah, absolutely. This is a super important area, of course, because, the processes that we do in the lab, often use plasticware. We go through a lot of chemical waste, but certainly in my lab, this is front and center is trying to find ways to actually reduce our carbon footprint, reduce the amount of plasticware that we're using, reduce the energy consumption through our -80 freezers. But it's a constant struggle because research is a process that uses up a lot of resources. But you don't want to do carbon dioxide research without being, having the best possible stewardship. So this is constantly a focus in our lab, is how to better our practices.
Thank you. I'll go back to the Q&A box. You described carbon capture and storage before you launched into a discussion of protein modification by CO2. It's important to emphasize that you don't have an alternative CO2 sequestration strategy, and climate scientists' insistence on reduction of fossil fuel burning to get us to a safer climate is as important as ever. It's a comment that you probably might want to comment on too.
So yes, I mean, trying to find carbon capture technologies is very, very important. And I think that investing in biological carbon capture is really going to be a major contributor to helping to change our industrial processes. So I couldn't agree more. This is a really active area of research, and it's being done more effectively right now in other areas of the planet. Unfortunately, in the UK and in places in Europe, there are several companies that are really ramping up biological carbon capture. And I hope in Canada we can move toward using more and more of these technologies in the coming decade.
Thank you. This is going back to your Indigenous perspective of learning. Can you give specific or concrete examples on how we can reduce emissions from an Indigenous perspective of understanding and interacting with nature?
Right. So I think that, yes. So ultimately, like in an Indigenous perspective is the idea of sustainability. And one example of that particularly applies to carbon and the atmosphere is the Indigenous practice of cultural burning. Okay, so what we're seeing in BC is every single year we have these major burns, these plumes of smoke that are given off by forest fires that basically are decimating large areas of forest. Well, traditionally, Indigenous communities realized this was going on, and in order to mitigate this, did the practice of cultural burning. So they actually burned during the shoulder seasons very small stretches quite strategically of the understory around trees in order to be good stewards and not create this system where these forests can simply just ignite. And so that's an example of how Indigenous practice has directly led to better forest stewardship that has reduced greenhouse gas emissions.
Thank you. Thank you so much. The next question is on something medical related. You were explaining earlier about how the brain responds automatically to detect CO2 in our body. Can this CO2 detection help us detect diseases or disorders much earlier?
Oh, I see. Yeah, so the system that's used in our brains is mostly about controlling our breathing rate, but it's not so much about detecting, bacterial infection or
Right.
this kind of thing. But
Okay.
you pose a really interesting question, because it's known that CO2 levels actually change innate immunity, so our immune response.
Mhm.
And so our immune response is directly impacted during bacterial infection.
Right.
And
Mhm.
so it's likely that there are sensors that are involved, you know, and it help to change that interaction that we have with different bacteria, but that's not yet understood.
Right. Right. Makes sense. Next question. What are some of the outstanding questions and challenges in this field?
Yeah, so it's a really broad field, but definitely within my area, the idea is this, that we understand, and I hope that some of this was hammered home through the talk, that CO2 has such an important role in organisms, but we don't actually understand what the heck it's doing.
Mhm.
And so in my field, that's like so important right now, is to develop new research tools that can actually enable us to study what this modification is doing on biomolecules and cells. And so that's where the focus of our research is really at, is improving our methods and better
Mhm.
detecting these sensing pathways in cells.
Mhm. Thank you. And related to that question, perhaps for any high school students in the audience tonight, would you have any tips to share to inspire them towards this field of study?
What I would say is I can only give my sort of background, certainly. I've long had an interest in science and what occurs, beyond what we can see. And that's what draws me myself into, the biomolecular sciences. But I was really along this pathway that was getting into studying detailed biomolecules and scientific processes. And I didn't realize until a little later on in my career that these can be applied to really important problems that matter to us. And that's why I pivoted and, stopped focusing so much on issues of human health and more on aspects of climate change more recently. And I think that that's one thing that I would say is just there's room for all types in academia, and there's room for all different types of focus in biomolecular science. And so what truly drives you, you can pursue that. In my
Mhm.
case, it was these climate change aspects. In your case, it might be something else.
Mhm. So the key is to identify what tickles you and what makes you really curious about something.
Exactly. And there's room to explore.
Yes. Next question. Is there evidence that increased CO2 in the atmosphere has detrimental or beneficial effects on cyanobacterial growth?
Yeah, great question. There certainly is evidence, and oftentimes from the cyanobacteria's perspective, it can be quite beneficial, because a warming planet and increased CO2 levels lead to an increased frequency of what we call cyanobacterial blooms, which is where they start to overtake our waterways and become very prevalent in the ocean. So from the cyanobacterial's perspective, this is very good, but from our perspective, these cyanobacteria can be quite toxic. So and they can also really disrupt the ecosystems by overgrowth in this way.
Mhm.
So I think that's an important message to hammer home as well.
Thank you. Next question. C4 plants have a much higher affinity for CO2 than C3 plants. Is there any plant to genetically engineer its CO2 fixing enzyme into cyanobacteria?
Oh, this is a really great question. So yeah, this is very insightful question. And basically, one of the things to bear in mind is that when we think about plants, they're kind of, they have these stomata that let the gas exchange in and out and let CO2 come in and out. But the special thing about cyanobacteria is that mechanism that I showed you where it can actually actively pump CO2 into the cell. And so really, if we could engineer plants to do this and to effectively pump CO2 into the cell and fix it, we could really improve plant growth. And there's a whole field in science that's trying to get at just that, combining cyanobacterial and plant cells in order to have more effective carbon capture in plants.
Mhm. Thank you. You mentioned earlier in your presentation that CO2 sensing can sometimes be poorly understood. Is there anything else we can do to facilitate a better understanding of this study, and what else can we do?
Yeah, so really, really good question. It's not just my field of research that really bears in on this question, and we have a lot of collaborators that are genetic engineers. So they can go into the actual genetic material of the cyanobacterial cell and make changes. And these changes can be very informative as to what the molecular pathways of CO2 sensing are. So we're combining our methods with people who are doing genetic engineering in order to combine these and get a much, much deeper understanding of where these CO2 sensing mechanisms are coming from.
Thank you. It's been a fabulous discussion so far, Dustin. I'll probably end with this last question because it does touch on sustainability, because that's ultimately the focus of your research. What drew you to working on microbiology that's focused on sustainability?
Oh, yeah. So yeah, I mean, my motivations for sustainability are really coming from this inherent desire to contribute toward that area in humanity, as I see it as so important. And I think that microorganisms really give a lot of potential, in part because, you know, if we look at plants, certainly scaling up a biological carbon capture is much more difficult in plants. But in cyanobacteria and different photosynthetic microorganisms, you know, we can readily control their numbers. They grow quite efficiently. They're very easy to make, as I say, these genetic changes to. So really working in microbiological and a bacterial model gives us a very effective model system in which to study the CO2 capture biologically.
Well, thank you. Thank you so much. There's so many other comments on the Q&A box, all appreciating your research presentation tonight, and everyone saying great talk, fabulous area of research. Thank you so much for sharing your time tonight in explaining all of this. We appreciate all the questions as well coming from the audience. Thank you so much for joining us tonight. Thank you so much, Dustin, for your time tonight. I hope you enjoyed it too.
All right, I did. And thank you to everybody for tuning in. It was a lot of fun.
Thank you. So that's it for tonight. We hope you can join us again for the next Café Scientifique session. It's happening. There's none for December, but we will come back in January. And you have that information on the slide that you see on your screen. It's January 30th. And as I mentioned earlier, because you are now in our mailing list, you will receive the Eventbrite registration information for this session and the rest of the sessions for 2024. Again, thank you so much, Dustin, for joining us tonight. And thank you, everyone, for always supporting this discussion series. We hope to see you in the new year in the next session. So again, have a great evening. Thank you, everyone, and take care. Bye for now.
Bye.